Welcome back to the deep dive. Today we're navigating one of the most complex tumors out there, diffuse large B-cell lymphoma, or DLBCL.
Yeah, it's the most common blood cancer, actually. But it's notoriously aggressive and incredibly varied from one person to the next.
And that variation, that heterogeneity,
Hmm.
that's the real clinical challenge, isn't it?
It absolutely is. It's what keeps oncologists up at night. While many patients respond well initially, something like 40% are either refractory right from the start.
Meaning the treatment just doesn't work.
Exactly. Or they relapse pretty quickly afterwards. And even with, amazing advances like CERAR T cells and bispecific antibodies, we're still struggling with a big chunk of those relapsed cases.
So the issue isn't just the cancer cell itself, it's the whole context, the neighborhood it lives in, what we call the tumor immune microenvironment, the time. Why has that been such a difficult thing to really understand?
Well, it largely comes down to how we've studied it traditionally. If you do bulk sequencing, you grind everything up and get an average.
Like 10% T cells overall in the sample.
Right. Which tells you almost nothing about where those T cells are or what they're doing.
Yeah.
It's like describing the average height of everyone in New York City, totally useless for figuring out what's happening on a specific street corner.
Okay.
And then even with newer tools like single-cell sequencing, which give you incredible detail about each cell, well, you still have to physically break the tissue apart to get those single cells.
So you lose the map. You lose the spatial context entirely.
You got it. You know what the cell is, maybe its state,
Yeah.
but you have zero idea where it was, and crucially, who it was interacting with. Neighbors matter.
Right. You lose the interaction, you lose the function.
Precisely. That interaction is where the mechanism happens, the suppression, the activation. So our mission for this deep dive is really based on a key study that tackled this head-on. They used cutting-edge spatial technologies on, I think, 78 DLBCL tumors.
Okay.
The goal was to actually map out these cellular niches, these functional units of interacting cells, and figure out how the local geography, the niche, drives what the tumor cells and the immune cells are actually doing.
So mapping the battlefield in real time almost. What kind of tech lets you do that? Getting single-cell detail without shredding the tissue. Sounds like serious hardware.
It really is. They essentially built two maps and overlaid them. First, highly multiplexed spatial transcriptomics using the NanoString CosMx SMI platform to see where nearly 1,000 different genes were being expressed, cell by cell, in situ.
In the tissue itself.
Yep. And then they added a special proteomics map using a technology called CODEX, looking at about 31 key proteins on the cell surfaces. Putting these together gave them this unprecedented view of cell identity, cell state, and location.
Okay, let's unpack this high-resolution map then. What did seeing things at this level reveal that, standard pathology just wouldn't catch?
Well, it let them get way beyond just saying tumor cell or immune cell. The analysis defined 19 really distinct cell types and functional states.
19.
Yeah. So, for instance, the most common type, no surprise, was the proliferating tumor B cells themselves, about 35% of all the cells they mapped. But they could also distinguish different types of plasma cells secreting different antibodies, like IgG or IgA, a main T cell cluster. Various types of tumor-associated macrophages, TAMs, like these interesting APOE plus C1q plus ones, and even the non-immune, non-cancer cells.
Right, the support staff you mentioned. For listeners maybe less familiar with how a lymph node is built, what are those non-malignant cells? What's their role?
Good question. So you have cells like follicular reticular cells, FRCs. They kind of provide the physical scaffolding, the structure of the tissue.
Okay.
And then you have things like high endothelial cells, HEVs, which are specialized blood vessels. They act like gatekeepers, controlling how and where immune cells enter the lymph node from the blood. These aren't passive players. They provide crucial signals.
So that structure, that scaffolding, leads us right to the core idea of this study, the cellular neighborhood, or CN. How exactly did they define a neighborhood in this context?
They shifted focus from just the individual cell to its immediate surroundings, its functional unit. They drew a circle, basically, around the center of each cell, about 24 micrometers radius, or 200 pixels in their images.
Pretty tight circle.
Very. And the cellular neighborhood for that central cell was defined as the collection of all cells, typically 10 to 20 cells whose centers fell within that radius.
Okay. So it's not just about the cell itself, but its immediate social circle, essentially.
Exactly. It's a difference between looking at one person versus looking at everyone interacting on, say, a busy street corner or in a small meeting room. That's where the action is.
Makes sense. And by looking at the mix of cell types in these thousands of tiny neighborhoods, they found they clustered into 7 common types. 7 stereotypical neighborhoods.
That's right. 7 recurring patterns of cell colocalization, or CNs. And importantly, the frequency of these CNs varied hugely between different patients' tumors.
Which one stood out as being particularly important in DLBCL?
Well, a few key ones. CN1 was heavily enriched in T cells. CN5 was this really dense core of tumor B cells, almost exclusively tumor cells packed together. That made up about 14% of the tissue they looked at.
Okay, the fortress.
Kind of, yeah. And then the most common one, covering over a third of the tumor area, was CN6. They called it diffuse. This was where you saw tumor cells really intermingled with a whole mix of different immune cells, T cells, macrophages, etc.
And that difference between CN5, the dense tumor core, and CN6, the diffuse mix, that sounds critical.
It absolutely is. CN5 is like the tumor stronghold, dense, restricted, maybe harder for immune cells to get into. But CN6, that's the real interface, the active battlefield.
Because the tumor cells are right there with the immune cells.
Exactly. When they looked at co-localization networks in CN6, the tumor B cells were right at the center, physically close to lots of different immune cell types. If you want to understand how the tumor is interacting with and likely suppressing the immune system, CN6 is where you need to look.
Okay, let's pause here for a sec. So we've gone from just counting cell types to seeing this intricate spatial organization. 19 distinct cell states arranging themselves into 7 common neighborhoods. This feels like a much richer picture.
It really is. And the study pointed out something important. Those older ways of classifying the microenvironment using bulk RNA sequencing, categories like inflamed or depleted, well, they only showed a pretty modest overlap with these directly measured spatial neighborhoods.
Suggesting the spatial map is much more accurate.
Definitely. The older methods were inferring proximity from a mixed-up soup of genes. The spatial mapping shows that inference wasn't always right, because location, actual physical closeness, is everything.
Which brings us to a key question then. If location dictates function, how does a T cell actually behave differently when it finds itself in, say, that dense tumor neighborhood, CN5, compared to when it's in an immune-rich area like CN1? Is it fundamentally the same cell just acting differently based on its address?
Right, because the initial analysis just found one big T cell cluster, C5T. But when they looked at where those T cells lived, in which neighborhood, suddenly their functions looked completely different.
Dramatically different. It's like giving the same person different jobs based on where they are. Let's compare 3 key addresses for T cells.
Okay.
First, CN1, the T cell rich neighborhood. Here, the T cells looked, well, healthier, more functional. They expressed markers associated with naive or memory states, had high levels of cytotoxic markers suggesting they could kill tumor cells, and importantly, low levels of dysfunction or exhaustion markers.
And their neighbors were helping them out. It seems like it. The main neighbors, these APOE plus, C1q plus TAMs, a type of macrophage, were expressing high levels of genes involved in antigen presentation, like MHC class II. So they were likely showing antigens to the T cells, kind of educating and supporting them.
Okay, so CN1 is a supportive environment. What about CN3, the myeloid-rich niche?
CN3 T cells looked activated but also stressed. They had high levels of cytotoxic markers too, like granzyme K and perforin, meaning they were trying to fight. Yeah. But at the same time, they showed high levels of exhaustion markers, things like LAG3, TIM3, 41BB.
So they're engaged in the fight, but maybe burning out.
Exactly. It suggests intense, maybe chronic activation, but they're starting to show signs of hitting an immunological wall. Dysfunction is creeping in.
And then there's CN5, that dense tumor fortress. Sounds like a bad place for a T cell.
The worst place, functionally. T cells found in CN5 were clearly suppressed. They had low cytotoxic markers and instead showed high expression of alternative dysfunction or inhibitory markers like ENT-PD1, TGD, and importantly, PD1.
PD1, the classic immune checkpoint.
Right. This neighborhood isn't just passively excluding T cells. It seems to be actively suppressing any that do manage to get close.
So what controls whether T cells even get into these neighborhoods? Why are they recruited to CN1 and CN3 but seemingly kept out of or suppressed in CN5?
Must be communication signals, right? Chemokines?
Precisely. It comes down to cell-to-cell communication. First, recruitment. Chemokines like CXCL9 and CCL5 signals that basically say, T cells, come here, were highly expressed in the healthier niches, CN1 and CN3.
Who was sending those signals?
Came from various cells, those helpful TAMs, the tumor B cells themselves in some contexts, and even the structural FRCs. But, crucially, those recruitment signals were almost totally absent in the suppressive CN5 niche. The keep-out sign was up.
Okay, so the door is shut in CN5. But if a T cell does somehow get in, there are mechanisms to shut it down. inhibitory signals,
Exactly. That's the second layer, the classic immune break, the PD-L1 to PD-1 interaction.
where PD-L1 on one cell binds PD-1 on the T cell and tells it to stop. Right? That interaction was significantly enriched in the suppressive niches. In the myeloid-rich CN3, the PD-L1 often came from the myeloid cells. But in that dense tumor core, CN5, the tumor B cells themselves were a major source of PD-L1. They were directly applying the brakes to any nearby T cells.
Wow. Okay, let's zoom back in on those tumor B cells in CN5. They're not just densely packed. The outline mentioned they were highly proliferative, almost mimicking a normal immune structure.
Yes, that was a fascinating finding. The gene expression patterns and the spatial arrangement in CN5 looked remarkably similar to the germinal center dark zone.
Which is?
The dark zone is a specific area in a normal lymph node germinal center where B cells undergo incredibly rapid proliferation and selection. It's characterized by high proliferation, high BCL2 expression for survival, and typically low numbers of T cells. CN5 mirrored many of these features.
And was there a specific communication pathway driving this dark zone-like behavior in the tumor?
There was. The key interaction seemed to be CXCL12-CXCR4. The FRCs, those structural cells, were found to be provided the chemokine CXCL12 within CN5.
Okay.
And the tumor B cells in CN5 expressed high levels of the receptor CXCR4. This CXCL12-CXCR4 signaling is known to drive B cell migration, survival, and proliferation. It was massively enriched specifically in CN5, suggesting it was a key driver of that dense aggregation and growth.
So the tumor cells are actively using signals from the microenvironment, from the FRCs, to build their own proliferative fortress.
It certainly looks that way. And interestingly, the tumor cells in the more diffuse CN6 niche seemed less reliant on this specific interaction. They were less proliferative overall.
Okay, quick recap then. We've seen that T cell function isn't uniform at all. It completely depends on the neighborhood. CN1 is relatively healthy and supported, CN3 is activated but exhausted, and CN5 is this actively suppressed proliferative fortress, almost like a cancerous dark zone driven by CXCL12, CXCR4 signaling.
Exactly. It really drives home the point that just counting the number of T cells in a biopsy isn't enough. You absolutely have to know where they are and who they're talking to. It's about functional ecology, not just cell counts.
That makes total sense. Knowing the location tells you the likely function. So we've got the general rules of this DLBCL architecture. But what about specific situations? Like, does a viral infection like EBV or the tumor's location in the body change these neighborhood patterns? Let's start with EBV, Epstein-Barr virus. About 5% of DLBCL cases are linked to it. Normally, the immune system should be targeting EBV-infected cells. So what does the time look like in these EBV-positive cases? Does it look different?
It does look quite different, compositionally at least. The study found that nodal EBV-positive DLBCLs actually had significantly more T cells infiltrating the tumor and correspondingly a lower percentage of tumor B cells compared to EBV-negative cases.
More T cells. That sounds good, right? More soldiers on the ground?
You'd think so.
I think so.
And spatially, this translated into an overrepresentation of the T cell-enriched niches, like CN1 and another mixed niche, CN7. So the T cells were definitely there and in the right kinds of neighborhoods, structurally speaking.
I sense a but coming. There's a paradox here.
There is. Despite being in these seemingly favorable niches and being present in high numbers, the T cells in EBV-positive tumors showed this really striking pattern. They had simultaneously high expression of cytotoxicity markers, like granzyme B and perforin.
Meaning they are trying to kill.
Yes, and very high expression of exhaustion markers, particularly HAVCR2, which encodes TIM3.
Ah, so they're revved up but also hitting the brakes hard at the same time. Primed but dysfunctional.
That's a perfect way to put it. It points to a state of chronic stimulation. The T cells are constantly seeing the EBV and tumor antigens, but this relentless engagement has driven them into exhaustion. They're present, they're trying, but they're functionally impaired.
And clinically, that specific state, that has implications for treatment, right?
Huge implications.
Yeah.
This primed but dysfunctional state strongly suggests that if you can release those brakes, for example, with immune checkpoint inhibitors like anti-PD-1 or potentially anti-TIM3 therapies, you might unleash a powerful anti-tumor response. It provides a spatial mechanism for why EBV-positive DLBCLs can be particularly responsive to immunotherapy.
Okay, that makes sense. Now, let's pivot from cause, EBV, to location. What about tumors in so-called immune-privileged sites, IPS, places like the brain, the CNS, or the testis, which normally try to limit immune responses to avoid damage? CNS lymphoma, for instance, is really tough to treat.
Yeah, this was another really interesting part of the study. You'd expect these sites to have few immune cells, right? That's the definition of immune privilege.
Right, limited trafficking.
But what they found in the 11 IPS DLBCL cases they looked at was surprisingly high T-cell infiltration. The T-cells were getting in. And spatially, they're mostly found in those intermixed neighborhoods, CN6, diffuse, and CN7, mixed, meaning they were right there, mingling with the tumor B cells.
So the privilege wasn't preventing entry, at least not completely. What were those T cells doing once they got inside? Were they suppressed like in CN5?
Actually, no, not entirely suppressed in the same way. They showed really strong signs of an ongoing active immune response. T cells within these IPS tumors had significantly higher expression of cytotoxicity genes, granzyme B, perforin again, and also higher markers of proliferation, suggesting they were expanding in response to the tumor.
So an active fight was happening, even in these privileged sites, but the tumors still grow, so there must be a catch, a counterbalance.
There absolutely was.
Yeah.
Alongside that high activation and proliferation, the T cells in IPS tumors also showed very high expression of multiple co-inhibitory receptors simultaneously, LAG3, TMM3, PD-1.
All the brakes being slammed on at once?
Pretty much. And the analysis showed high activity of both the PDL1, PD-1 pathway and another important one, galectin-9 TIM3 signaling. So the picture in IPS DLBCL is one of intense inflammation and immune activation that is being very actively and perhaps effectively suppressed by multiple checkpoint pathways working together.
Which, again, points towards potential therapies.
Exactly. It suggests that even though these sites are privileged, the issue isn't a lack of immune response, but rather an overwhelming suppression of an existing response. This makes IPS DLBCLs potentially strong candidates for immunotherapy, maybe even combination therapies targeting multiple checkpoints like PD-1 and TIM3 to release those brakes and let the activated T cells do their job.
It's fascinating. Whether it's EBV driving chronic stimulation or an immune-privileged site trying to dampen inflammation, the end result seems to be these activated but exhausted T cells. The tumor often wins by controlling the brakes, not just by hiding. And seeing that spatially is key. #tag outro. So, to wrap this all up, the big takeaway from this deep dive seems to be that in DLBCL, the spatial architecture, where cells are and who they're talking to in these distinct neighborhoods, is really the fundamental decider of immune function and ultimately patient outcome. It gives us a much more precise map than we ever had before.
I think that's exactly right. It moves us beyond just cataloging mutations or counting cell types toward understanding the functional ecosystem. This spatial characterization provides a roadmap for intervention that those inferred signatures from bulk or even single-cell RNA-seq just couldn't provide reliably.
And it opens the door for more personalized approaches, presumably.
That's the hope, absolutely. Imagine looking at a patient's biopsy, mapping its neighborhood composition. Is it dominated by that suppressive CN5 core, or the inflamed but exhausted CN6, or maybe the EBV-like pattern? And then choosing the therapy most likely to work in that specific microenvironment. Maybe you need to recruit T cells into CN5, or maybe you need checkpoint blockade for CN6 or EBV cases.
And looking even further ahead, can we get this level of insight without needing these complex, expensive spatial platforms every time?
That's the ultimate goal. The idea is that with enough of this high-resolution spatial data from studies like this, we could potentially train sophisticated AI models and deep learning models. These models could then learn to recognize the subtle patterns associated with these different neighborhoods directly from a standard, digitized H&E stained slide, the routine pathology slide that's done for every patient.
Wow, so democratizing this complex spatial information.
Exactly. Making this kind of nuanced microenvironmental insight potentially available for guiding treatment decisions for everyone, everywhere. That's long-term vision.
Well, this has been incredibly insightful. It really underscores that in the complex world of cancer immunology, context is king. Who you're standing next to profoundly shapes what you do. Thanks for joining us for this deep dive.
My pleasure. And maybe we can leave you, the listener, with one final puzzle from the study to think about. We mentioned that some tumors evade immune attack by losing MHC class I genes, things like B2M or HLA genes, basically making themselves invisible to certain T cells.
Right, a classic immune escape mechanism.
Well, the researchers observed that DLBCLs carrying these MHCI loss mutations actually tended to have more of that diffused inflammatory T-cell infiltrated CN6 niche. Now, why would a tumor that has evolved a major way to hide from the immune system preferentially hang out in the most inflammatory immune cell-rich neighborhood?
Yeah.
It kind of suggests the tumor isn't just escaping the immune system. Maybe it's actively managing or even manipulating that inflammation, something to ponder.